Alright, so we're going to call this video Unit 10, Section 2. I know it doesn't say that. This is about mitosis. So, you may have learned in middle school that mitosis was cell division, and that's not technically correct. Mitosis is nuclear division. It's only the division of the nucleus. Let's highlight that. It's only the division of the nucleus. That's no good. Okay. The rest of the cell is divided in something called cytokinesis, which we'll learn about in a few minutes. So it's the division of the nucleus of all of your body cells. Every cell in your body that actually divides, except for sperm and egg cells. Now, red blood cells, white blood cells, and nerve cells, they don't divide, so they don't go through mitosis, technically. So, if you divide the nucleus, you're basically dividing the DNA. You're taking the amount of DNA an organism has in one cell and dividing it into half. So each new cell will have half the DNA as the original cell, but only temporarily. Make sure you understand that. Only temporarily. If you combine cytokinesis, which occurs right after mitosis, together both those make up the M phase. We talked about that in the last video. Mitosis has 4 basic phases or sections, but I'm going to divide the first phase into actually 2 subphases. So that's going to give us 5 phases. Mitosis lasts about 2 hours in humans. So here's a simple diagram of mitosis and what happens in each of the phases. Now I've divided prophase into what I'm going to call early prophase and late prophase. That's because prophase lasts about an hour of the 2 hours, half the time. And there's a lot of events that occurs in prophase, so I'm going to divide it into 2 to make it just a little bit easier for you to commit to memory. So if you look at the bullets over there, prophase is half mitosis. Second bullet, the nuclear envelope. In order for the nucleus to divide, the nuclear envelope, which is kind of like that fence around the nucleus, it has to go away. It has to gather away, so it starts to dissolve. It starts to break down. And then once you remember the parts of the nucleus that we learned a few units ago, the nuclear envelope, the nucleolus, and the DNA, all those comprise the nucleus. The nucleolus has to go away, so it breaks down too. And then the chromatin does something kind of neat. The chromatin, which is the DNA, but chromatin is in thin, stringy, long strands. They're invisible in a light microscope. If you want to think of it, chromatin, if you want to incorrectly call it chromatin, that will remind you in your brain that chromatin is very thin. Chromatin actually coils up, which means it gets fatter, and it gets shorter. And these shorter, fatter pieces of DNA are called chromosomes, a word you've heard of, and they're actually visible in a light microscope. You can see all these dark areas right through here. These are actual chromosomes. So let's look at what prophase means. Pro means before or first. It's the first phase. That's why they call it prophase. The nuclear envelope breaks down. The nucleolus breaks down. That's 2 events. The chromatin condenses and coils in a chromosome. That's 3 events. These little things called centrioles, which are perpendicular microtubules that are sitting like side by side right outside of the nuclear envelope. They're sitting right next to each other. At the beginning of prophase, they begin to move apart to opposite ends of the cell, which we call the poles, like North Pole, South Pole. So these centrioles, which occurs right outside the nuclear envelope, or they sit outside the nuclear envelope, during mitosis, prophase, they start to move apart, and they go to opposite ends of the cell. As they're pulling apart, between them are these long protein fibers called spindle fibers, and they form a spindle apparatus between the 2 centrioles. And this occurs from the beginning of prophase all the way to the end of prophase. So late prophase, these spindle fibers have forms, and these chromosomes which have condensed, now the chromosome attaches to a spindle fiber. And it attaches to the spindle fiber at a location on the chromosome called the centromere. There's a diagram of this a little bit later, so we'll see it in just a few minutes. Okay, by the end of prophase, the spindle apparatus is fully formed, And asters, more little microtubules, project outward like in a starburst pattern. That's what aster means, by the way. It means star. These asters form around each centriole, and you'll see that in a few minutes. And this only occurs in animal cells. And then at the very end of prophase, very late prophase, these spindle fibers with the chromosomes attached to each spindle fiber begins pushing and pulling through a mechanism that scientists totally don't understand yet. But they see, you can see them move. They start being pushed and pulled. Next phase, metaphase. Meta means middle. What's not the middle phase? Well, actually, it is the middle phase of the 5. But what the middle means is these pushing and pulling of the chromosomes, they get eventually lined up right across the equator or the middle of the cell. So you can see them lined up right across here in one line. And you can very faintly see these spindle fibers, these long fibers stretching from almost one end of the cell to another end of the cell. So what I'm highlighting here are kind of like where the centrioles are. So the one event that occurs in metaphase, chromosomes line up across the middle. Next phase, anaphase. Ana means opposite. So at this point, when the chromosomes are nice and lined up at the middle, these spindle fibers begin to pull towards the poles, towards the centrioles, and it rips the chromosome apart into 2 pieces that we call sister chromatids. Yeah, I know, in other words, it sounds very similar. These 2 sister chromatids are genetically the same. They contain the same genes. They may have different versions of the genes, and we'll talk about that when we get to genetics, but they have the same genes. So the centromere that was attaching to the spindle fiber, it actually divides into 2 pieces, and the sister chromatid goes to one half of the centromere, and the other sister chromatid goes to the other half of the centromere, and they get pulled apart, and they migrate towards opposite ends of the cell. This is all done by the spindle apparatus. In this picture, you can actually see where the centrioles are. Let me change colors on this. Red. Okay. There's where the centrioles are. You can't really see the centrioles, but that's where they are. And then these outward projections that you can sometimes see here, these are all the asters. Now, they think asters serve as an anchoring point in the cytoplasm so the spindle can do its pushing and pulling, but they're not totally sure about that either. Next phase, last phase of mitosis, telophase, and telo means the end. At this point, these sister chromatids have been pulled, and they arrive at the poles where they stop. So they're going to, what's going to happen is, and you can see this, the cytoplasm starts to pinch in. Now, even though it's not totally divided yet, it's pinching in. It's coming towards each other, towards a middle point. I'll draw a couple of arrows. Okay. This is actually like the first sign of cytokinesis. But what telophase is defined as is everything that kind of happened in prophase. Everything that disappeared or everything that formed does the opposite. So chromosomes, which thickened and coiled, now they unwind and relax back into chromatin. The nucleolus went away. Well, the nucleolus comes back. The nuclear envelope broke down. The nuclear envelope comes back. So everything that happened in prophase, here's a word for you, unhappens in telophase. Now that is officially the end of mitosis. Right after mitosis, there's an event called cytokinesis. Let's go back just a second. And this pinching in of the cytoplasm, if you can imagine, it comes on together. And there's not a good photograph of this, so here's a drawing. The pinching in totally completes, and the 2 brand new daughter cells separate. Okay, each smaller than the parent cell, and each with half the DNA the parent cell had temporarily. So cyto, remember from a couple of minutes ago, cyto means cell, kinesis means to split or divide. So it's the splitting of the cytoplasm, or simply the splitting of the cell. And it's not part of mitosis. It's a totally separate event after mitosis. So here's some diagrams of some of the things I just mentioned a few minutes ago. Like here's a close-up version of a chromosome. Here is one sister chromatid, and here's the other sister chromatid, and together it makes a chromosome pair. And each chromosome pair is attached to a spindle fiber. That's this right here. And then right here in the middle is the centromere, part of the chromosome, and that's where it attaches to the spindle fiber. So we humans in a cell, we have 23 of these structures, or 46 separate chromatids, 23 from mom, 23 from dad. So here are the centrioles and where they're located in some of the phases. In interphase, they're side by side outside the nuclear envelope. In prophase, they're moving apart towards the poles. In anaphase, they're at the poles. And in telophase, they're in separate cells. Here's one pair up here, and here's the other pair down here. This is a close-up of the centrioles, and they're simply microtubules. We learned about what those were in the cell structure chapter. And then here is what we're showing you, the spindle apparatus. The entire spindle apparatus is the centrioles plus the spindle fibers. This happens to be metaphase. They're all lined up across the middle. And this drawing, which really looks like almost like a photograph to a microscope, is showing you all the phases of the cell cycle, except I don't think they show you telophase on here yet. Many of these cells are in interphase, and there's a couple in prophase. There's at least one, I think, in metaphase, maybe almost metaphase. I see some anaphase. I want you to see if you can pick out which one they are. Which one can you identify the 4 phases? Except, like I said, telophase is not on there. All right, so if you want to look on the internet, there's a lot more pictures and a lot more videos and animations than just this. You can see mitosis actually occur in an animation. You can see what happens, all that pushing and pulling of the chromatids, of the chromosomes, and the splitting apart of the chromatids. So don't just limit yourself to what's on this video. Explore, go to Google, go to YouTube, and explore. Lots of good stuff there. That's the end of this video.